So in my first slide, I actually superimposed upon our website photo a picture of a microbial community, and you'll notice that there's a lot more variety in cell shape and size there, because I do want to emphasize the fact that we're talking here about microbial communities. And in my case, I'm going to talk exclusively about recovery of genomes from microbial communities and genome-resolved studies, which we refer to as genome-resolved metagenomics. So I think I can skip this because Curtis just showed it. This is a statement of the goals of the workshop, and continue on to talk about why metagenomics and why genome-resolved metagenomics. So this is probably very familiar to everybody in the room, but of course microbes are really important in the world around us, the world inside of us. They mediate most of the processes we care about in terms of biogeochemical cycles in soil, in water, in sediment. Most of them are uncultivated. This is especially true for the natural environment, not so true for the human environment. Genome sequencing methods have revolutionized the field. They provide new ways to investigate microbes in context of the geochemical data that we are obtaining from these sorts of systems. And they enable functional studies, meaning, for example, proteomics. So I want to stress that natural environments are complex, but actually they're variably complex. Some are much simpler than others, and the challenges really scale with complexity. But of course, new bioinformatics methods are critical. So this I used to just sort of motivate my own thinking to you. It's a diagram from a paper published about 18 months ago by Kelly Wright and et al. From our lab at the time. And it shows how we put together the kind of data that we obtain from natural systems to inform modeling efforts. And this is a GWAS model that's being developed by Owen Brody at LBNL. So the modeling I'm not going to talk about. I'm going to talk about what goes into the model, which basically is information about what kind of organisms exist in the environment, the nature of the metabolic capacities, and from the predictions we were able to make, we want to be able to assign roles to organisms. So for example, we can identify certain organisms that break down the resources in the environment, which exist in a variety of different forms, to produce byproducts which drive other microbial metabolisms, which in this natural system, this is a subsurface sediment, carry out various geochemical transformations of interest to us. So you can see here that we're linking into quite a number of different cycles: iron, ammonium, nitrogen, hydrogen, iron again, and sulfur. I also want to point out on this slide that there's two organisms here with these kind of enigmatic looking names, OP11 and OD1. So these are organisms that come from phyla, or maybe even superphyla, that have no known isolate representatives. It's clear that those types of organisms play very important roles in natural systems, and we cannot build models for natural environments and their functionings without incorporating all of the organisms, not just the ones we can grow in the laboratory. So here you can see we have explicitly incorporated the role of these organisms as best as we are able to understand it into our overview of microbial community function. So this is what we're trying to get to. This is obviously a pretty simple system, but one that's, I think, emblematic. So to amplify on this point, this is a picture. Actually, it's two pictures, one of the bacterial domain and one of the archaeal domain. And I like to say now this is kind of old. This is way back from 2013. This is a very rapidly moving target. But the main take-home point here is that there are many phylum-level groups in domain bacteria and archaea. And the majority, if not the vast majority of them, lack isolated representatives. So, clearly, it's important to develop and apply methods that can detect these organisms and also provide clues as to what they're doing in their environments. So one of the methods we all hear a lot about is 16S ribosomal RNA gene surveys, and I think it's probably a good time to take a moment to say what metagenomics gets us that the 16S doesn't, and I'm sure it will become apparent likewise that the opposite is true in other ways. That there's things you can do with 16S that you can't do with metagenomics at this time. The biggest concern I have at the moment is it would appear from comparative studies where we don't have PCR bias that these kinds of surveys can miss 20% on average of all the organisms in the environment. And I want to point out that sometimes it misses 100% for some lineages. So we must know going in with surveys that are based on an expectation of sequence that we're going to have gaps in what we see. They also have limited taxonomic resolution, which is great for some things. But for certain kinds of questions, it's really important to be able to say something about what a species is doing or a strain is doing. I'll give an example of that in a moment. And they're also fundamentally limited in their ability to provide information about abundance because there's not a single copy gene. Just to reinforce this point, this is a study conducted joint with the Joint Genome Institute and funded through the ETOP program, where we're comparing actually single-cell genomics and metagenomics. Metagenomics, it's genome resolved, and I'll go on to the methods in a moment. But you can read the details here. It's a little bit of a detailed analysis, but the take-home point is the results are very consistent. In another system, subsurface, something like 20% of the organisms would effectively fall through the gap if we're using these kinds of approaches to survey microbial community composition. And to amplify the point about why we need to go to species or strain level. This is just one example. I guess you probably have your own examples, but let me show you this one. This is an organism called Gallianella. It's a very well-known iron oxidizer. It's very difficult to grow in the lab, though there are isolates. There is one genome, this one Gallianella capsidiferiformans ES2. This organism, for which there is an isolate and a genome sequence from the isolate, cannot reduce nitrate or nitrite. But lo and behold, an organism that's almost identical at the 16S level, as shown by the little inserted tree here, actually has that capacity. In fact, we can identify a 50-some kb block of the genome sequence that encodes all of the capacities for nitrate and nitrite reduction, including all the accessory proteins and so forth. So had we made a prediction based on a genome that, for all intensive purposes, is almost identical to Gallianella capsidiferiformans, we would have had the wrong result. And it turns out this was very coincidentally fortuitous, because the realization that the genome had these capacities coincided with an experiment in the field where nitrate was added to the subsurface and this organism bloomed. So right away, the environmental experiment was telling us this was the case, and we can provide an explanation from the genome analysis. Okay, so what do I mean by genome-resolved analysis? We go into the environment, we take samples of the whole microbial community. As I mentioned before, of course, they're mixed species consortia with varying levels of complexity. We extract the DNA, we sequence it. We assemble it, which is itself an interesting and important topic. We bin the data, and I'm going to spend quite a bit of time talking about the binning methods, by which I mean we assign the genome fragments to genomes. We then analyze the genome sequences. In the ideal world, in cases of interest, we curate the genomes. In some cases, we curate them to completeness. It's not always possible. It's frequently not possible. But if we can, it's important to do so, because the value of a complete genome is substantially higher than the value of a draft. Predict genes, predict functions. We want to resolve genomes for all of the relatively abundant organisms in the community, and we can routinely do this now for certain systems. And by that, we mean genome-resolved metagenomics. Well, I wanted to go back just over 10 years now to where this started, at least started for us, where we were working with a model microbial community with very low complexity. This was an acid mine drainage biofilm, and we went on to study these for now 20 years, but this was the first time genomes had been recovered from a natural environment. This was doable because at that time, Sanger sequencing had become high enough volume that we could get 75 megabases worth of sequence from the JGI. We were ecstatic. And I think we eventually argued 135 megabases of sequence out of them in the end. This is expensive sequence. But nonetheless, this worked because we had high dominance of our microbial communities. The most abundant organism in the sample often was something in the order of 45% of the sample. So it was possible to get draft genomes, very good draft genomes, as it turned out, for 2 of the organisms, and less good drafts for about 3 others. This remained kind of on the sidelines for a very long time, because people would say to me, you can do it in acid mine drainage, you'll never do it anywhere else. And it remained true for a long time, actually. It took the advent of high-throughput short-read sequencing to make it possible to begin to adapt these methods. Of course, many people are doing this, not just us, but for us and others to do this, to a whole variety of other ecosystems where, of course, it will find application. And it does work. That's not to say there aren't challenges. So this is just an example of some of the systems that we're working on at the moment. Many of these systems are with collaborators, David Relman, for example, working on dolphin mouths and seal mouths, and I'll talk a little bit about human infant. But you'll notice that these systems cut across the boundary between the environment and human health. And I think that's very natural and appropriate, that we ask questions that are common across all environment types and don't partition human microbiome away from the rest. In fact, when I was asked to be involved in this workshop, I thought that's actually one of the really neat features. that we're bringing together people from across these disciplines and fields and asking questions that do cross-cut the various boundaries between the sciences, but also are very relevant to a whole wide spectrum of real applications of societal relevance. So I'm going to talk mostly, very briefly, about assembly and touch on the topic of curation, but I want to talk mostly about binning. So just on the topic of assembly, this is something, again, we've been interacting with the GGI group quite a bit, and Susanna's here somewhere, I think, and she's the person who's been working most closely with us. But the question is, what kind of assemblers to use for metagenomics, to short-read metagenomic data? And the answer is it's not seemingly, unless you guys know something I don't know, and I'd be happy if that's the case, but really any good answer. One of the real limitations, I would say, with a lot of the assemblers that are around at the moment is they don't scaffold. And RDBA-UD, which is used by many people to do metagenomics assemblies, does scaffold. And you can see the consequence of this if you look at the statistics put together by Brian Thomas. You can see, for us, the most important thing is we have lots of large contexts, which were then available to be curated into really high-quality genomes if you use this scaffolding step. This is really vital. The problem is IDBA-UD makes errors. Maybe all assemblers make errors, but these errors are actually very systematic, but they can be corrected, and it's my perspective at the moment, in the absence of a better solution, that better to use the assembler that scaffolds and to have an algorithm that corrects the errors than to not scaffold. And actually, the reason why the scaffolding is most important in some ways is because it is a binning step. If we can put the pieces of the genome together, we don't need to bin them. So when it comes to binning, we can identify a number of different sources of information about how to make a bin, how to assign the fragments that come from a single genome into a bin that represents the genome of that organism, as shown here. I'm going to talk a little bit about phylogenetic profiles because I think this is really, really important, especially for human microbiome research. GC content, abundance level of organism in a single sample, basically the sequence composition, including codon usage, and then the abundance level across multiple samples. It's probably pretty easy to understand that these things in combination can give you a very, very good approach for binning in some systems. But let me go into it to make sure it's clear. So we usually take our contigs and we profile them upon assembly after prediction of the open reading frames by comparing each open reading frame to a database. And I'll talk about the database in a minute because that's actually kind of important. But we basically say, where is the best hit for that protein in the known biology world? And we can see here that we're tracking, this is going phylogenetically down to a species level, that many, many, many of the proteins on this contig have a best hit to the same species, represented here as green, Clostridium perfringens. And so we would profile that as Clostridium perfringens. We're not saying it is Clostridium perfringens. We're not requiring that it be identical, but just saying that that's where it's leaning. And so this is very useful because it means we can collect together those contigs that share that characteristic. As we approach things of greater novelty, for example, this case, we can see that in the sample there is an organism that's bacteria that is in the phylum Actinobacteria, extracted here. This is this wedge here, in the Actinomycetales, but the species-based hits are inconsistent. The genus-based hits are inconsistent. You can see that we're hitting all over the board, so we profile this as the order Actinomycetales. So with this approach, GC and coverage alone, we were able to analyze a collection of human microbiome samples very, very quickly. This is an example of some of the data we got, and I put this up to really emphasize that the findings make the effort worthwhile, that we would have got the wrong answer had we just used 16S, or even perhaps not analyzed the data in detail. So one of the abundant organisms in these infant gut samples were these are infant gut, by the way, and it was a study that we were looking at necrotizing enterocolitis. The details so much don't matter here, but this is Enterococcus faecalis. When we looked at the sequences of a whole variety of Enterococcus faecalis from a whole variety of different infants, we found, by comparing the samples across time, this is the reference sequence for infant 3, that all the samples, all the sequences basically were the same within the same infant at different time steps, nucleotide level identical, with some exceptions, This is another case where that would be the black bars indicate SNPs. So basically we compare the sequence from infant 3 to infant 6. We can see, yes, it's Enterococcus faecalis, clearly, but there are sequence changes that make that strain distinct. So these would look the same at the level of 16S most certainly, but in actual fact the sequences are different. And this was really important because we were looking at transmission of an organism in an intensive care unit. So we want to know, is it the same or is it not the same? In fact, in most cases the strains were the same within the infant over time, but you can see here early and late for infant number 5, we can see a strain switch. And in fact, we often see this sort of strain switch associated with antibiotic administration and sometimes with other health considerations. So, critical part about this study, I think, was that we were able to analyze these data very quickly. And in fact, the binning for the samples was done in a time frame of minutes. We have, this publication came out just recently, and I didn't put the reference on it. It came out in eLife in 2015. It's on the next slide. We can see here we can document all the strains in all the infants, and we can compare the strains in the samples, and this is a heat map. So this shows this strain, this particular strain 6 of Enterococcus faecalis became variably abundant and passed through the red bar, which is the antibiotic treatment, so it was antibiotic resistant. What we found from this analysis, other than the fact that the sequencing is now the same step, is that, in fact, well, I'll get to that what we found in a second. Let me just show you one more thing. This is basically a time series of samples from a single infant. We can see in detail the community composition. This accounts for on the order of 90% of the reads, sometimes much more. We can see that basically the list of organisms present in this infant are more or less a laundry list of all the bad guys that are out there in the hospital environment. We were trying to explain why this infant developed necrotizing enterocolitis, which happened at this stage. There's the antibiotic administration. And we can track different organisms. In this case, number 3 would be Citrobacter. Citrobacter strain, very similar to KTE32. Across time, we see it pass through the period of antibiotic administration. We can see the same thing is true for Enterococcus faecalis, but in the case of the Veillonella, this organism was eliminated. So basically, we were able to go to the metabolic repertoire predicted from the genomes and show that actually organisms with a lot of antibiotic resistance did in fact persist through the treatment, whereas those did not persist. So what did we learn from this? Well, we learned a lot of things, a couple of things that we knew already, and I won't go through those, you can read that. But basically, contrary to our expectation, this was a study supposedly of an outbreak of NEC in the neonatal intensive care unit. Contrary to expectation, we found essentially no strains were shared amongst any of the infants. So there's a lot more strain variety out there than we expected, and certainly no 2 infants that became ill developed this disease, which we believe to be attributable to a microbe that was transmissible within the intensive care unit. And clearly that would not have been apparent. So basically, we would tend to believe that these infants were colonized by just basically a lot of different pathogens, and probably other factors determined that these infants became ill. So coming back to the topic of phylogenetic profiling, it works great in the human microbiome, especially in the infant human microbiome, though we've also done this with adult gut samples. It doesn't work at all well when you turn your attention to natural systems, particularly complex natural systems. So this is a study site in Japan, just as used as an example here. Here we're phylogenetically profiling the whole community using the method I mentioned previously. We can see bacteria, don't know, probably viruses, archaea, in terms of the breakdown at the domain level. Within the bacterial domain, we can see that a large fraction, maybe 1/3, is, maybe more like a half actually, is only profiled at the phylum level. So together here we're grouping a whole bunch of different organisms that just look like bacteria, but we don't know what they are. So needless to say, this is our unknown diversity. These are our microbes that have not been studied previously, and so we cannot recognize them when we detect them with metagenomics. So the good news here is that once we increase the quality and coverage of our database, the phylogenetic profiling kicks in. So I think this is actually very optimistic, because as we move forward and collect genomes from single-cell genomics, from metagenomics, and from isolates, and we add them to our internal databases, all of a sudden things become apparent. It's just one piece of information, but it can be extremely useful in narrowing down the taxonomic affiliation of fragments to put them into genomes. So, and this is an example we're putting in now, a set of TM7 genomes. TM7 is, again, one of these candidate phyla. And all of a sudden, we can see it's a TM7. It doesn't look a lot like anything we've seen before, but it's information. So I would posit that one of the things that will become very useful to the research community will be a well-curated set of reference data sets, reference genomes, which are near complete, if not complete, and we actually know what they are. And this is really important because sometimes things get into the databases from various sources that we actually can't trust the phylogenetic classification for. So we need to agree upon a method to do that, but basically we need to know if we're using it as profiling what these organisms might be. So it's very important, I think, to think about building such a database. I'm not going to introduce emergent self-organizing maps. They're basically just a clustering method for which we group fragments together, and they're very pretty. They're periodic, and this is the period of the map. But basically, there's just a statistical method to analyze sequence composition or other statistical information. And we use this a lot in binning. In this example from Joanne Emerson's PhD research, she was looking at Crystal Geyser, which is actually a JGI ETOP site now. And I'll talk about it in a moment, again, I think, in more detail. But in the very first data set, she had a very small amount of sequence information. She had a few dominant organisms. In this case, the tetranucleotide composition essentially alone was enough to bin the data, but of course more powerful when augmented by GC and the phylogenetic profile, the phylogenetic information that I just talked about. And so you can basically have confidence in our bins. We can basically make the bins by any method we want, any algorithm you have. And you can then ask, are they good, by creating maps of some sort with various inputs and then projecting onto them the answer that you've come about from those methods and see if they really look great. So Crystal Geyser is okay for the dominant things. Human gut's easy for the most part. But a lot of samples, a lot of systems, it just doesn't work. And so other kinds of information need to be brought to bear. And one of the things that's proved to be most powerful is information about abundance patterns of organisms and thus the fragments of their genomes across samples. I don't know if this is effective, but I've tried to sort of illustrate this as a set of samples. You can see this organism blue changes in its abundance, therefore its genome fragments should change in the same abundance pattern over the samples. And that's a signature of all the fragments that belong to that genome and distinct from this one. So we use this, and as do others, in combination with sequence composition and, of course, the other information being GC content and phylogenetic profile for binning. And this can be done in a variety of different ways. We have our own algorithm. Other people have different algorithms. I think at the moment we're still uncertain which ones work and when, but certainly this is a promising approach overall. They can also be used in an iterative way, and this is an incredibly complex slide from Alex Probst, but basically you should take away from this is that we have lots of sorts of information. We want to be able to make the predictions of the bins. We want to be able to validate the bins. If we don't get validation, we want to go back and refine them. And when you go through this kind of iterative approach with all the methods that you have on hand in a data series, you can get the majority of genomes from the samples, like hundreds of genomes, literally. And just to give you an idea of what a baddie sum looks like, it doesn't really matter. I don't even know why he calculated a 1 and 2 frequency, but basically this would be absolutely useless in terms of grouping together fragments. But if we add in now just the abundance information from just 7 samples, we can see we get very strong boundaries, which would imply that all the points that fall within these areas, which represent genome fragments, belong to the same genome and distinct from the one in the next area. And so this seems to work extremely well, and this is just basically a projection onto the map to show the identity of the organisms that were binned in this data analysis. Okay, so one of the things I'm really interested in is to make all this high throughput. So we can struggle through pulling our genome out, refining it, and it takes a long, long time. But really, for this method and this approach to become useful, we need to be able to do it for many, many samples, and we need to be able to do it relatively effectively and quickly. So coming back to the tree of life and the domain bacteria, I wanted to point out to you that, at least in this construction and supported by, I think, all of our subsequent calculations, there is part of the tree of life that groups together, for which there are no isolated representatives. Now, we call the ones with no isolated representatives candidate phyla, and so this would be a radiation of candidate phyla, and so we call it just simply the candidate phyla radiation and identify it as a major feature on the tree. So I'll show you some statistics in a moment, but from one series of samples, we were able to get just over 800 genomes, including 8 complete genomes. And this is shown on the left, and I would would say that it has a long way to go to be as good as it needs to be, but basically this is a clustering taxonomically of this radiation, but this is also metabolic analysis that's high throughput to get a sense of what these organisms do and don't do. And I should say that, perhaps just more for fun than anything, that this set of samples were obtained in a sort of special way. We took groundwater and we put it through a 0.8 micron filter onto a 0.2 micron filter, and then caught what came through the 0.2 micron filter. Now, those of you who filter water know that the 0.2 micron filter is technically sterilization, but clearly it's not, because this entire sample series was based on what went through a 0.2 micron filter. And this is not unique. Others have reported this too, including from 16S surveys in one case. And we can image these cells and get a sense of what they might look like. And basically, we can step back and then look at the commonalities across this radiation, knowing that they're small cells, they've got very small genomes, which is actually why we can get the genomes complete. They don't have repeats, including they only have one copy of the 16S. They've got all sorts of interesting features which you can, this is actually published now, which you can look at if you're interested to know. But basically the take-home point is we can really start to fill out the tree of life, including organisms that have not been cultivated. And we can do it with pretty darn good genomes. So I would agree, before you criticize me, that our metrics for genome completeness aren't ideal, and this is something we probably want to put on a list of things to talk about. But nonetheless, based on our best ability to determine at the time, we think these are on average about 91% complete. So this is over 800 genomes, and there's a few others I've thrown in here as well. So this is progress, but not enough, because there is another frontier that has been kicking our butt, to be completely honest with you, and that's soil. I kind of hoped it would be easy, and sediment seemed relatively easy, surprisingly easy, so it lulled me into a false sense of security, but that's not to say we're not making progress. Soil probably is the last frontier. It's incredibly heterogeneous, and there's a lot of strain variation, and that really complicates our assemblies. Nonetheless, we're having a crack at trying to make sense of these kinds of environments, and through the use of spatial time information, which is really important. We don't have nearly enough of it probably ultimately, but we have in the order of 12, I think, samples, plus tetranucleotide information, plus everything else we can throw at it. We are starting to be able to get reasonably good genomes. And this you can read as basically, these are all ones, unless they're darker. These are single copy gene inventories, which sort of indicate to us that the genomes are not too bad. And you can see here the ESOM constructed with this information starting to look like we can bin out good genomes. And they are novel. These are not things we've seen before for the most part. I didn't show you the profiling. So from my experience, the take-home point at the moment is that some systems are really easy, and we can bin out genomes of bacteria, archaea, phage, plasmids, no worries. And as we get to more and more complex environments approaching soil, the challenges become more and more significant. But we do need methods that work across ecosystem types. So I didn't have time to make a slide for this, but I just put one statement here, and I want to just speak to it. I don't know if you remember back in the early 2000s, there was a big debate between Tiger and JGI about whether or not we should have complete genomes. Some of you remember this. And, you know, I don't know anybody really ever agreed with anybody, but the truth of the matter is, practically speaking, it wasn't possible to get the kind of high throughput for genomes and have them to be closed and complete. But I think the time's come to turn around and say, you know, probably Tiger were right all along, this Venture Institute as it's now known, and we do want complete genomes, and we have to just suck it up and work out a way of making that happen. And I think with these integrated long-read technologies and more sequencing and better informatics methods, that can become a reality, because all the arguments that were made then are still true. You know the size of the genome, you know what isn't encoded, and so on and so forth. So it makes a lot of sense. So in the last couple of minutes, I just want to comment that the genome's all well and good, but you need to be able to work out what that genome is encoding. And if you've got hundreds or thousands or tens of thousands of genomes, you better have a way to do that simultaneously and efficiently. So this is a cell cartoon put together by Cindy Castell. It probably took her a year to do after she had a really good, closed, complete genome. And it's very detailed and a lot of biochemical modeling and so forth. This is great, because I think we really threw everything at it we could, but we can't do this for 100 or 1,000 genomes. So we need a way to do that more effectively. Our approach at the moment has been to capture metabolic prediction information as we go through the data analysis pipeline, to create what we call lists, where we search each data set for certain functions or certain genes of interest using a variety of different methods, And at the moment, I have to confess, our approach is mostly blast-based, though we do use other information. We're transitioning to a new way at the moment that I think is going to be a lot more robust. It's based on HMMs. But I think it's a start, and this is the kind of direction we need to be investing in. The idea then is if we have one genome, we can make a list of search terms, and we can basically categorize what's present in the genome to make a starting point for it. With this, it would be manual analysis. Is it really a SOX-C gene, or is it really something else? Is the annotation correct? And that now becomes back into the trenches and slow. But we need to be able to make that fast. And so basically we need to be able to scale to hundreds, or in this case we're dealing with 3,000 odd genomes. And the last thing, second to last thing actually, I wanted to make a comment about is that there's a lot of other information that becomes available to us once we have this kind of capacity. We have information from many samples. So we're going to ask questions about what is the pattern of distribution of this organism across these samples? Does it correlate with another organism? Does it correlate with a geochemical condition? And why? We're positioned to ask this, but we do need ways of being able to track the abundance patterns, and we just do that by read mapping. The answer to that is easy, probably, though I'm sure there's some nuances, but how to find statistically significant correlations so that we can probe why we see these organizations, why these cohorts appear in the environment and across environments. And of course, the first thing we need to do, if we've got data series, sample series across a watershed, for example, is to be able to dereplicate the genomes, and that's proven to be an interesting problem in and of itself. But ultimately, this is what we want to find, who tracks with whom and why. And I really think this is pretty much my last slide. I think in terms of an opportunity, this is something of a cartoon, but this is the opportunity that's in front of us right now, and that is to build a new tree of life. It's been a long time since we saw a really good three-domain tree of life that represents most of life. Certainly that's never happened, but now we're positioned to, with all of the genomes that are coming out from many, many research labs and many different approaches. And so what will it really look like when the dust settles? This is going to be a very interesting time. I should point out that don't trust the placement of eukaryotes here. That's just a root. Of course, that's a very controversial topic, and I do not wish to comment on the answer to that question. But I will make the point that eukaryotes are also kind of important too, and we probably need to be scaling our methods for metagenomics and community genomics to take into consideration, well, not just eukaryotes, but also, not just microbial eukaryotes, but also large eukaryotes. So I started off, I guess, kind of banging on 16S a little bit, but let me just back up and say what we can't do, that 16S can do at the moment, is deal with scale. And I was trying to think why. Well, I think we can scale our informatics methods to do a lot of what we need to do, to deal with hundreds or thousands of samples, but I'm not sure we can pay the library costs. So I really like to see some developments in reducing the library costs so that we can afford to make hundreds or thousands of samples. And maybe JGI can comment about that. There may be some tricks I don't know about. But this at least working with commercial facilities is quite limiting. I commented already about complete genomes and microbial eukaryotes. So I'm going to close now and just throw up some acknowledgements for people in my research group, and then our collaborator at the JGI, Susan Anna Trench, who's here, actually, and our funding. And I'm sorry if this presentation wasn't quite what you wanted. I know making excuses is not a good thing, and like the dog ate my homework, as Mary said to me earlier on, is a good excuse, but actually this is really true. The rat ate my power cord. And I should thank Mary for rescuing me last night with basically a loaner power cord. Okay, so thank you, and I'm not sure what the format is now, Curtis, but do you want questions? Or?
There's so many questions, but I guess I'll start with one, which is, you mentioned this factor of, there are other factors about why babies might get NEC because you're seeing different pathogens colonize those babies. And for this particular meeting, I think it might be interesting to delve deeper into that, to try and connect what's happening microbially to what's happening in these, let's say, nuclear genomes or metabolomes or transcriptomes. So what do you think is the way to get to those other factors?
Okay. Oh, looks like they want me to use this one. Okay, so this is an interesting, let me just back up and say that the reason we did this study is because there was an outbreak in the NICU. So I want to also say that the reason we're studying NEC, doing microbial community analysis, is we believe because there are outbreaks, in part, that this is a disease that's caused by microbes. So my conclusion about this, I don't know how to go forward is a different question. My conclusion about this is really that these babies are in an environment that's clean to death, and it's just enriching for these really bad set of players. And probably, my guess is that it's other factors of the infant health that really trigger whether or not they become ill, if in fact it's a microbe that causes it in the first place. How to study it in future, I mean, we're still dealing with small statistics of small numbers. I think that the one thing that is clear is that the strains are not the same. We looked for plasmids as well. We couldn't find plasmids or phage that would account for this. But more data would obviously be a really good idea as we move forward.
Thanks.
Jill Lugros back here.
Yeah.
Thank you very much both for the talk and for your help in organizing this. I appreciate it. So this is a naive question. It appeared from the soil, one of those soil slides that ran past, that there was a time response in that under different environmental circumstances, different water input, there were different responses of the system. And my question is, are there now perhaps some techniques from those who are very knowledgeable about geospatial analysis, they're geographers often, temporal GIS kinds of things, that might be useful in teasing apart some of the environment genomic interactions?
So I love that question, I have to say. So I didn't actually show very much data from soil, but that study is designed to look at a pulse of carbon released from soil after the first fall rain event. And there are big shifts in community composition and changes with depth as well. But I think I can best address that, in my mind at least, by turning back to the Rifle SFA at LBNL, which has focused previously at this site called Rifle in Rifle, Colorado. Some of you will know of it. Some not, obviously. But the mandate has been given to us to scale to the watersheds. And how do you go about trying to do this, what we're trying to do at the watershed scale? And the answer is to bring in geospatial information. Absolutely, that's exactly what we're doing. And to work with geomorphologists who are geologists who believe, well, no, who certainly do have an understanding about how systems work and how they evolve. And to have that intersection guide the choice of sampling locations so that we can take a systems within systems approach. And that's what we're basically rolling out. And to do that, we still have to scale the number of samples to hundreds, if not thousands of samples. But I think it's doable at the hundreds of samples level. So it's going to take another round of development in terms of methods to make that possible. But I cannot let this go back to doing 16S, because that's what the alternative would be, to be able to deal with something the scale of a watershed, I think.
Okay, Karen Lloyd, I'm at the University of Tennessee, and I'm wondering if you have a good, or what your preferred solution is for determining phylogenies of different genomes with different subsets of genes. I have trouble finding the common core often. What's your solution?
So you're asking me at genome completeness, or we talk about phylogenetic classification?
Phylogenetic classification, when the genomes are not complete.
So
as a universal way.
That's a great question. So we've taken a couple of approaches, and I mean, I'm sure other people should just jump in and give their answer, because that sounds like a really good topic for discussion. But we've made the case that concatenation of ribosomal proteins works quite well. And so we've chosen to use co-encoded proteins, and actually we do that. It's terrible for genome completeness, but it's great because there's no binning step involved. So basically we find a fragment that carries and we require 8 of 16. And we find that phylogeny, if you can get it, and you can't get it for all the bins, but 8 of 16, it's not too bad, gives us a phylogeny that's very consistent with 16S. And I think that is gaining acceptance as a method to do this. There are other, we've looked at RPS3, which is just another single copy gene, and calibrate it with 16S to get a sense of the divergence. But that's really basically what we would advocate at the moment. Does anybody else want to comment on this? Or it seems like a really interesting topic.
My interruption of you was in the Brown paper, you guys found that one very common ribosomal protein is not present in any of your new phyla.
That's right.
Which is a little shocking.
Yeah. Well, I mean, and we looked really, really hard. Yeah. There's actually a variety of different ribosomal proteins that are absent in those organisms. And their biology is just really different. They're so interesting. But yeah, so we don't rely on one, and we certainly wouldn't rely on one that's not, university present. I think Matt had a question.
Yeah. Matt Kane from NSF. Jill, the advances you brought to bear are all, they're huge advances, but they're focused on still genomes, complete genomes, cells, species, and that model of biological interaction. And I'm wondering if there aren't questions, for example, about ecosystem function that would be better looked at from a new model of just looking at metabolism and genes in the environment and not so much worried about which genomes they're packed up into.
So I strongly disagree with that approach.
Yeah.
And I think it's really important that functions come in packets that are organisms, and one organism may have one capacity, but it's linked to another set of capacities. And if you're going to understand how the system's functioning, it's a cell that responds, not part of a cell. So I think it's really important to keep the whole set of capacities in view, and that's what you lose if you just profile metabolic function. You don't know which function is in which organism. So that's been tried. It is kind of like an earlier approach. I think it's not the way to go, for the reason I just said.
So what about the situations where you've got a large number, hundreds of genomes that are similar, but the function of interest is only in some of them. And so knowing an organism is present doesn't actually tell you the function is present.
So ideally, you'd know that all of the organisms are present, because this goes down to the strain level. So you'd know you have organisms that have this capacity and don't, and be able to track their response over time. And certainly they will respond differently. Organisms, subtle differences will respond differently over different conditions. So I think you can see that. And I mean, in terms of doing hundreds of samples, the question that you may be walking around is, can we actually bin genomes out of hundreds or thousands of samples? And the answer, I don't know. But I think so, because the one thing we've learned about binning is that series-based data is valuable. So if I have 100 or 200 or 500 different samples with organism abundance patterns, I think the algorithms will bin the data instantaneously, or do a pretty darn good job. So that's my hope going forward anyway. Yeah.
Hi, Morgan Langelo from Dalhousie University. Sort of follow up on that question. I mean, and to push it a bit further, do you think, though, besides trying to predict, say, genomes to metabolites, if there's some way that we can say these sets of genomes are likely to produce these metabolites, right? So there is the possibility that we could identify every single strain out there, but it seems like there's a probability around all these things, right, that this set of genomes or this micro community will produce this metabolite. I'm wondering if there's a little bit of flexibility, not completely going to say it's just a bag of genes, but if there's, just going a little bit beyond sort of reconstructing one genome and saying it's this one genome, but it probably could have this gene that, is laterally transferring or something. And have you thought about sort of a model like that besides just hard, fast genomes?
Well, I mean, I'm holding the line, but I'm ready to compromise, I guess I would say. I mean, I would say, just to follow up further on my answer to Matt, and I think maybe it's relevant to throw it out here too, that I believe the modeling approaches are calling for genomes, not for functions. If you want to build a really robust model of how community works, you should have the capacity to have those functionalities predicted. So when you talk about metabolites, do you mean proteins or do you mean small molecules? Because those are very different balls of wax. I mean, one of the things we did a long, long time ago is show you can do strain-resolved proteomics. So we can resolve down to the sequence level for proteins. Of course, metabolites, it's a totally different problem. Were you thinking one or the other or both?
I was sort of mixing them together, but I mean, yeah.
Yeah.
I was just trying to get away from that sort of, is it really possible? Does this genome really exist sometimes is what I wonder. I mean, maybe it doesn't. It exists in a cell, but I'm just wondering if it's better to think of it as slightly cloudy around that cell and that there's genes that
I think when
fluctuate.
it comes to soil, we're just basically going to have to do that. I mean, Gematomonadites is one of my favorite at the moment. You get 8 or 10 strains, and you really can't separate them. And so, at some level, there will have to be some compromise in that regard. And so it may become a probability, as you say, probability that this, I think that's what you're getting at, that this Gematomonadites is on 85% probability or something that these cells have that function and are expressing it. Yeah, I think that's a valuable sort of an idea. Yeah, for sure.
I'm Mary Lipton from Pacific Northwest National Laboratory, and the comment, this is more of a comment than a question, in going back to the discussion we had, is that we have to think of the genomes not as an end in itself. It's the beginning of a whole process for characterizing these systems, and there's a whole suite of analytics that comes after the genomes, and if you have the binning, you can actually start putting metabolomic and proteomic and transcriptomic data into some sort of context to then look at organism interactions and the functions that are present. So yes, while the genome may not tell you whether that function is actually present, the follow-on analytics will give you an idea of what is active, when it's active, and the suite that we have to follow on utilizes this kind of data. So we have to think of this as a starting point, not as an end in itself.
Absolutely.